Okay, so we're going to start record. Okay, so now, so what's the in terms of gene mutation? What we're going to study here? If not mistaken, part of the parts of these topics already covered before. Okay, so we need to explain the gene mutation. In this case, we are talking about the gene mutation. So we do know that what's meant by gene? Gene is particular sequence of the DNA, okay, that code for a specific polypeptide. So today we're going to look at the gene mutation, not the chromosomal mutation. Okay, we're going to learn about chromosomal mutation next topic. Okay, but for this topic, we only focus at the gene level. Okay, gene mutation occur by three methods here. Three methods. It can be substitution. So from the word substitute, so one nucleotide substituted by another nucleotide, and then deletions means that the nucleotide have been deleted, and insertions. Insertion sometimes we also call it as an addition. Okay, so it means that we add some bases or nucleotide, okay, or the base pair of DNA, and outlines how these mutations may affect the phenotypes. Okay, how it can change the phenotype. Okay, so we're going to use one example here for albinism, a disease called albinism. We're going to relate the gene, enzyme, phenotype with respect to the thyroxinase, a gene called thyroxinase involved in the productions of the melanin. So how dark or how intense our skin pigmentation is because of this melanin, because of this melanin. So we're going to look at how the melanin productions is lower because of the mutations. And then the last part is more to the very factual part. We're going to look at the effect of mutant allele on the phenotype in the following human conditions. So albinism, sickle cell anemia, hemophilia, Huntington disease. So this 4 is in our syllabus, and I'm going to discuss another 2, okay, which we're going to use it a little bit in the next 2 topics. One is another 2, we're going to call it as a cystic fibrosis, called CF. We're going to look at this cystic fibrosis, and another one we're going to look at this is the phenylketonuria, PKU. So these 2 I'm going to include it, but it's not in our syllabus, okay, for this topic. So we look at what is the gene mutation. So first, we started with the mutation. When we talk about mutations, mutation is a random and spontaneous change. It can be a very random, out of sudden, okay, because of the exposure to the mutation. It can be because of the DNA repair. It can be because of the DNA replication. So during this time periods of times, you might cause the number of DNA, structure of DNA changes. Okay, so change what? It changes the sequence of the nucleotide bases of a gene. So in this form, we call it as a gene mutation. Okay, it's a mutation at the gene. Okay, or it may cause the number or structure of the chromosome. Basically means that we do know that humans, we have 46 chromosome, right? So it might change the number of chromosome. Basically means that chromosome number increase. For example, in Down syndrome, 21. Okay, 21. So 21 basically means that you have extra 1. A person have 21 means extra 1 chromosome 21. So it causes the Down syndrome. Okay, so what are the causes of the gene mutation? What are the causes? So which event may lead to the gene mutation? The first one, spontaneous mutation. This one is out of our control every time our DNA replication takes place, particularly during DNA replication. Why this thing can happen? So for example, if a template here, or a parental strand, A, C, C, G, A, C, for example, and then during the synthesis of the new strands, so we add T, G, correct one, G, C, T, G, so making the complementary strands. Okay, so this one because of the DNA replicates semi-conservatively. Okay, but it's ideal to have this, but sometimes you may see that DNA replication during the process of DNA replication is not perfect. Okay, so instead of T, okay, G, maybe here they add T. So you see that mutation actually take place. Can you see that? So in this case, we call as a base substitution. We add we replace, again, by rights with G, we replace it with T. So this is because of the DNA replication. It may not be, what we call it, even though the error is low, but you still have a little bit here and there, the error, okay? So, or this is the parts that you need to know, this one, okay? Spontaneous mutations and during DNA replication, or the mutations due to the error-prone replication bypass of naturally occurred DNA damage. So due to DNA damage, so DNA tends to, DNA replications tends to bypass those errors. For example, okay, so something happened at this region called error. Okay, so during DNA replication, they add one by one, and they skip here, they continue. So it means that this part will be deleted. Can you see that? This part will be deleted. So this is because of the naturally occurred DNA damage. And then during DNA replication, the DNA polymerase tends to ignore the particular or bypass the particular DNA damage. Okay. Or sometimes error induced during the DNA repair. So just now we say that what happens if we do have this kind of mutation? So at the low rate of the mutation, our body still have times, have the mechanism to do the DNA repair. Okay, we still can repair our DNA. But the problem here is, if the error introduced during the DNA repair, then we have a problem. For example, in this case, by right, I should add, remove T. Okay, remove T and add G. But in the process of this, error actually introduced during the DNA repair. So it means that during repair, we make a wrong one again. So just now T, by right, put G, right? But now it becomes A, for example. So still got the error. So this kind of error is during DNA repair, okay? Or the last one that we need to know is induced mutation. Now, induced mutation basically means that induced by what? By the mutation. So from the name mutation basically means that the agents that cause the mutation. Now, again, it's not really true if we say that mutation is the agent that caused the mutation, but it's caused the mutation at very high rate. And this high rate is how high? It's high until our body repair or DNA repair mechanism have no time or fail to repair this DNA mutation. So it means that this DNA will going to skip the checkpoint and continue to be replicated, or even until the mitosis will be distributed to two different cells. Okay. So understand, in this, so mutation arises, right, but spontaneously at very low frequency. Okay, normally in our body currently is very, very low frequency because the chemical instability of the purines and pyrimidines. Okay, due to what? Due to the DNA replication. Okay. Then, in induced mutation, alteration of a gene after it has come into contact with the mutagens or environmental causes. Okay, so what's mutation? Mutation actually is it can be either physical or chemical agent that change the genetic material, okay, usually DNA, by increasing the frequency of mutation. Means that our body will have the general or normal way of the mutation, or normal rate of mutation, but with the mutation, it can be a physical mutation or chemical mutation. So therefore increases the frequency of the mutation. So this table actually shows you some example of the chemical mutations. You do not need to actually go and memorize it. No need, just for your informations only. So don't get traumatized. Oh, do I need? No, this is for your informations only. We do have a different classes of chemical mutations. Okay, so one of these we're going to learn in a little bit detail the usage of this in the gene technology is ethidium bromide. This name will again appear. Okay. Now, what are the physical mutations? So physical mutation, you need to know we have the ionizing radiation, for example, alpha, gamma ray, okay, X-ray. Those are the ionizing radiations, or generally the UV can cause the mutation to take place. So just need to know the name of the example physical mutations such as ionizing radiations or UV. Okay. So in terms of the mutations, we have two form mutations. We can have the germline mutation. This is more dangerous. Why I say more dangerous? Because it can be passed on to the descendant through the reproductive cells. For example, in the gamete. Okay, mutation in gametes, or mutation at the reproductive organ, for example, in ovary, in the testes. Those are very, very dangerous because this kind of mutation, if continue, are going to be inherited by the descendant. It means they can pass it to the offspring. Okay. Now, next we call it as a somatic mutation. Somatic mutation basically happen at your somatic cells, which does not involve in the reproduction. For example, skin cell. Mutation at the skin cell basically cause a skin rash or skin cancer. So this kind of mutation, it won't transmit to the descendant. So it takes place only at the somatic cells, not transmitted, okay, to the descendant or to offspring. Okay. So next, we're going to look at this. This is very important. You must know this. I put a star here, the gene mutation. So we have three modes of mutation here. So one is a base substitution. Second is a base insertions or base additions. And the third one is a base deletions. Now, here, I want to emphasize very, very important point here. When you talk about DNA mutation, we always mislead by what? The word base. We always use the word base substitution, base insertions, base deletion. But what is the definition of mutation? We have to word it in this way. When the DNA mutation is a change in the nucleotide sequence. Even though we always use the word base, but actually we don't change the base only. Are you clear? So what's the difference between the base versus nucleotide? Okay, so what's the difference? Then you understand why we say change in the nucleotide sequence rather than base sequence. Because during the process of the DNA mutations, then actually we change the nucleotide. So base basically this. So if you look at the structure, use a different color to show you guys. So we do know that DNA consists of the deoxyribose with the phosphate group. So what is the base? Base is this. So we don't change the base. We don't change the base. We don't alter the base, but we alter the entire nucleotide. This is nucleotide. So the base together with the deoxyribose and also the phosphate group. So we change the entire nucleotide. Even though in the name we use the word base substitution, base insertions, and also the base deletions. Okay. So this is the mode of mutations, and what is the effect of the mutation? So here I'm going to introduce few terms. We're going to look at it now. First, we call it a silent mutation. Okay. Second, we have missense mutation. Third, we have nonsense mutations. Okay, not nonsense or missense. Right? Not nonsense. That is nonsense mutations. And the last one we're going to look at is a frame shift mutation. Okay, we have silent mutation, missense, nonsense, and frame shift. So what are the difference here? So let us look at the detail. Okay, because this one you need to know. So what are the difference? So first I start it with this. So silent mutation. So what means silent mutation? Silent mutation only caused by base substitution. It must be base substitution. And if you still remember, you recall back our DNA triplet code. Okay, we do have, okay, DNA triplet code. 3 digit or 3 letter code for one amino acid. Clear or not? Are you clear? But it's not only one code. We have more than that. Okay, we have more than that. So it means that we do have another code, also code for the same amino acid. Are you clear? You make code for same amino acid. So in this case, means that when you substitute it, for example, base substitution take place. Okay, let's say let me find out one. DNA triplet code. It's about, okay, don't need to remember, always remember. Now, let's say, for example, in this case, TCT. This DNA triplet code for serine. And DNA mutation take place. We substitute what? We substitute the T here with the C. So it becomes TCC. Mutation take place, right? So it's a base substitution. So what kind of mutation? It's a base. What is the method? It's a base substitution. That result in what? Can I see that? TCC also code for serine. So means that in this case, the base substitution caused the or result in a silent mutation. So what means silent mutation? Means that the effect of the mutation cannot be detected in the polypeptide level. So means that you go to the polypeptide and check, nothing wrong. But you check a DNA, oh, mutation take place. So this kind of the mutation we call as a silent mutation. So it means that you don't see any effect, no effect. Okay, you will see that no effect on polypeptide. But when you check the DNA, you can see that DNA nucleotide change already. Okay, sequence is altered. Okay, so this is means by the silent mutation. Okay, it means that it's like something keep quiet, you can't see it. Okay, so in this process, if I draw it out, the entire polypeptide still remain the same. So no mutation. With mutations, two of them remain the same. No shortening, nothing, no special thing happen. Okay, silent mutation. Okay. Now next, we're going to look at the second one called missense mutation. Missense mutation also only caused by what? Caused by base substitution. It cannot be because of the addition or deletion. It must be base substitution. So what does this mean? Okay, let's see. The original one, the original base, let's say for example, we have TTC. Okay, TTC code for phenylalanine. Okay, phenylalanine. And base substitution take place, removes, okay, instead of C, now base substitution become A. So now mutation take place, become TTA. So TTA will code for what? TTA will code for the leucine. So you can see that there is a change in the amino acid already. So, but the entire length, very important in missense, only one amino acid. In this case, only one amino acid affected is substituted. Okay, so in this case, phenylalanine. In this case, so phenylalanine substituted by leucine. And because of this, then you can see that the polypeptide may not fold into the precise three-dimensional shape. Are you clear? It's not the original shape anymore. So the proteins or the enzyme may lose the function. It depends how severe. If let's say the mutation didn't take place at the critical region, for example, active site or binding site, protein somehow still can function, might decrease in terms of affinity, may decrease in terms of the catalytic reactions. The Km value may change, Vmax may change due to this, a slight change of the amino acids. So this kind, if we compare, so we still get back the original length of polypeptide. So the length still remain the same. So means that if let's say this case, this is the original one. So the mutation take place, so you get something like this. Okay, got one different, but the length still remain the same. So this is the one that change in one amino acid. So cause what we call as the missense mutations. Are you clear? It causes the missense mutation. What is the mechanism? What is the method here? The method is because of the base substitution. Okay, so this is the second one. Now we look at the third one, nonsense mutation. Sorry. So what's mean nonsense mutation? Nonsense mutation is because of the mutations. Okay, what is the method? It can be base substitution. It can be base additions. It can be base deletion. So means that 3 types of them can contribute to nonsense mutation. So what's mean nonsense mutation? Nonsense mutation because of this base substitution, base addition, or base deletion accidentally introduce stop triplets. Okay, stop triplets into the gene. Okay, before it by right it should be stopped, then it introduce it. Okay, so let's say for example, this triple coat, DNA triple coat, and then we do have one here, TAA. Okay, TAA, and then we have another triple coat. Sorry, 3AC. Okay, TAC for example. Okay, and then triple coat, and then we do have the last one. Okay, TAA. So this 3A is the original stop triplet. Are you clear? It's original triplet. Okay, so what will happen here is if base substitution take place, change the C to A. So means that this one become stop triplet already. Can I see that? TAA is a stop triplet. So means that by doing this, it will cause the polypeptide to become shorter. So the idea here is after the point, during this is the point of mutation. Okay, point of mutation. So anything after this cannot be synthesized already. Cannot see that, cannot be translated because nothing. Okay, stop triplet is introduced in the midst of the gene, rather than at the end of the gene, but at the middle of the genes here. So it means that our polypeptide actually become shorter. So it becomes shorter already, definitely they're going to lose the functions. Okay, or just now I say that you may add what? Add the A here. Can I see that? Or you may delete the C, then it becomes, okay, let's say for example, okay, TAC. Triplets. Okay, so what happened here is we add, what do we add? We add A here. So this is a base addition. Okay, this is a base addition. Or it so happened that TAC here is A. T for example. Okay, so what happened is if let's say deletion take place, the reading frame become TAA. Can I see that? So this one also as a stop triplet. So in this case, you can see that 3, okay, base additions, base deletions, and base substitution, all of them have the ability to result in this nonsense mutation. So the idea of nonsense mutation means that the synthesis of the polypeptide in the ribosome will stop prematurely. So it means that even though it's not the you won't be able to get the full length of the polypeptide. So it depends where. If let's say towards the end still okay, maybe it can function, but it's at the middle or at the beginning, okay, quite beginnings of the polypeptides, then we can see that the effect is very, very severe. Okay, so this is why I say that missense mutations versus nonsense mutation. Question asked before, what are the main differences? Missense, you still get back the same original length, only one amino acid change on it. Okay, but for nonsense mutations, you're going to get what? You're going to get the shorter polypeptide. Okay, it can miss a whole chunk of the amino acids. Okay, due to what? due to the introductions of the stop triplets or stop codons, okay, in mRNA. And the last one, also very, very severe, is called frame shift. Frame shift mutations only can be caused, okay, can be caused by base additions or base deletion. Now, what's mean frame shift? Frame shift basically means that you're going to alter the reading frame, 3 digit by 3 digits. You're going to alter the reading frames, okay, of the remaining of the DNA after the mutation. So what's this mean? Now, try to look at this. So we do know that we always have 3 digits. Okay, the coding, 3 digits, 3 digits. We read it this way. Okay, the triple quote. But if we delete here, we delete here. So the reading frame now become like this. The 3 digit. Can you see that? So means that any amino acid after the mutation, so it will be a wrong amino acid already. It's a different amino acid sequence already. Or different amino acid sequence. Can I see that? So because of this, that entire length, okay, after the mutations, entire length, you can see that they are no longer, okay, they are no longer a correct one. So therefore, the entire polypeptide confirmed cannot be used anymore. Are you clear? So this kind of mutation is called frame shift mutation. Why? Because we add something, we add a nucleotide or we delete a nucleotide. When you do this, the reading frame actually wrong already. The ribosome will read it. Because how to read it? Because you deleted here. Hence we deleted it here. Okay. So for example, you can see that if I have a T T T C C C G G G, for example, A T T and so on. Okay. So because of deletions here, then my reading frames become A TTC, CCG, GGA, TT, and so on. So it means I can see that the reading frame all wrong after the mutations. Okay, so in this kind of the situations we call as a base deletions and base additions. Okay, but something special here. As long as sometimes we don't add or delete 1, we add maybe a 3. Okay, so we call it trinucleotide deletions or addition. Then the is severe. But what we can see here is with trinucleotide deletions and additions, the effect not as severe as 1. So means that if I ask you, if I have 15, okay, nucleotide deletion versus 5 nucleotide deletion, which one more severe? Both of them are severe. Both of them are severe. 15 nucleotide deletions versus 5 nucleotide deletions. So which one more severe? In this case, actually, this is more severe. Why? Because if 5 is not a multiple of 3. Are you clear? As long as it's not multiple of 3, what will happen here is you will have frame shift. But 15 nucleotide deletion, why no frame shift? Because if 15 nucleotides, what will happen is like this. Okay, 3, 3, 3, 3, 3, 1, 2, 3, 4, 5. So 15 nucleotides. So this is 3 nucleotide, 3 nucleotide, 3 nucleotide, okay, 3 nucleotides and 3 nucleotides, a triplet. So I delete here 1, 2, 3, 4, 5. There is no frame shift. Can I see that? No frame shift. So you only have 5 amino acids missing. Okay, remove. Okay, only 5 amino acids removed, but the remaining one still correct. Can I see that? So even though it means we say that it's still severe, but not as severe as 5 nucleotides, which it will cause the frame shift mutation to take place. So it means that the reading frames are wrong. Okay, so let me put it in these table forms to summarize. So we have the base substitution, we have the base additions, and the base deletions. So basically addition, deletions, they belong to the same category. So what is the effect here? So silence mutation. So this one only can be caused by substitution. Additions cannot, deletions cannot. Okay. Second, we have the missense mutation. Only change one amino acid. So means only substitution can, additions, deletions cannot. Okay. So third one, we have the nonsense mutation. If nonsense mutations, then 3 of them can. Why? Because they can these 3 mechanisms can introduce, okay, the stop triplets or stop codons, making it shorter. Okay. The last one, frame shift. So frame shift mutations only can be caused by additions and deletions. Substitutions cannot. Are you clear? Frame shift cannot. Substitution, no way for substitutions to cause the frame shift mutations. Okay, so understand the concept, then you will be able to figure out. Okay, So next, we come back to our notes, introduce some other terms. Okay, so what has been the base substitution? So base substitution basically is involved with the exchange of single nucleotides for another. So most common one is the transitions. Now it's been transition. Transition basically we exchange purines for purine, pyrimidines for pyrimidine. So it means that we don't alter it. Okay, so it's quite common. For example, we have the T here. Okay, so by right, when you have the new one, we need to add A. But because of the mutation take place during the DNA replication, instead of A, because A is a purine, okay, A is a purine, but we get G. A and G, both of them are purine. Can I see that? Okay, so when you exchange purine to purine, basically means that the distance of the DNA still remain the same. Correct or not? Okay, so a bit of revisions here. If you forget, we do have the purine. DNA pairing, base pairing, we also pair purine with pyrimidine. Why we need to pair purine and pyrimidine? Why we cannot purine and purine? Why we cannot pair pyrimidine and pyrimidine? It's because of the distance. Okay, because of distance. Purines actually 2 ring, so larger. Pyrimidine only 1 ring structure, so smaller. So always 2 with 1, 2 with 1. That is the principle of the base pairing. So under purines, we have A and G. Pyrimidines in terms of DNA, we have the T and C. Are you clear? Okay. So if original is A, we put it as G. So for example, just now I say that if here is T, by right we have to add A. This is correct. But in the transition mutations, then I have T. Now I add in, instead of A, I add G. Now, because we do know that A and G, both of them are purine 2 ring. T is 1 ring. So it means that the length or the diameter of the DNA still remain the same. So this kind of mutation are more common. Okay, are more common. This kind of mutation compared to another one's called transversion. Now, why transversion is less common? Because it's going to alter the length or the what we call it, the distance between, or the diameter between the 2 DNA polynucleotides. Okay, now look at this. So now we start the original one. Here's A. So by right, A, we need to add T. Okay, so A is a purine. Okay, 2 ring. So T is a pyrimidine, 1 ring. Okay, Now, if let's say now I don't add T in, okay, we add G. So what will happen here is if you add G instead of T, means that you change, sorry, okay, if you change the pyrimidine now into purine structure. So if you change the purine to pyrimidine to purine, what will happen here is the DNA now become bulging. Because touring and touring, right? By right, we have 2 and 1, 2 and 1, 1 and 2. So it's standardized, okay, your DNA. But now this is touring, making it become bulging. So this bulging easily, okay, can be detected by DNA repair mechanism. So it means that mutation did take place, but DNA repair mechanism can easily detect them and make it back to normal, change it, edit it. Okay, so in this case, when you do an edit, in this case, you will be able to repair the DNA back to the original one. Okay, so this is what I say that transversion is less common. The effects less common because we have DNA repair mechanism. So what are the effects here? So we have a silent mutation, which code for the same amino acids. Therefore, you won't see any difference, okay, in the polypeptides. We have the missense mutations. It code for different amino acids. So it may result in a non-functional polypeptides. Are you clear? Okay, or less functional, not so effective now. All nonsense. Why? Because the code for a stop triplet or stop codon may truncate. Truncate means shorter, shortens these polypeptides. Okay, nonsense. So next, base insertions or additions, okay, may result in the frame shift mutations or nonsense mutation. Just now I say that. So frame shift mutation because the reading frame shifted. Oh, sorry, I don't know what actually happened here. Sorry. Okay, a shift in the reading frame in this case. So it may result in a completely different translation from the original. Okay, you can see that's a totally different after the frame. Okay, so the reading frame after the mutation, the reading frames now change already. Okay, or nonsense mutation as usual because of the introduction of stop codons that truncates the polypeptide. Okay, so base deletion, same thing. Okay, base deletion, same thing. One or more nucleotides are removed from the DNA. So again, you can result in the frame shift mutations and also the nonsense mutations. Okay. So with this, I stop here first. Do you guys have any questions? You can ask me, okay?